Oh my.
Says it's starting. It says it's starting. Hey, Kai, before we actually get the show started, before we get the show on the road, do you want to come up here and say hi as Justin is going and getting something very quickly? Hey, everybody out there.
Hi.
Kai wanted to say hi.
Hello.
I mean, you no, don't do that. Oh God. Okay. Climbing on the child climbing on me. Okay.
Hi.
What is your name?
Kai.
Okay. How old are you?
8.
What did you want to talk about tonight?
Global warming.
Why?
Oh, I just really think we should start focusing on global warming a bit more than we actually are currently.
How can we focus on it? What can people do?
Well, maybe use their lights less, like when it's daytime, turn off unless it's like a really dark area and you can barely see in there.
Mhm. Maybe use LEDs
Yeah.
and lower
Yeah.
lower electric requirement devices.
Try and buy electric cars maybe.
Mhm. And why is this important to you?
Just because
Talking to the mic.
everybody's going to die.
No.
And everything living on the planet.
I don't know if everything living there are extremophiles.
Mostly.
Yeah.
Mostly
All right.
everything.
Well, we will talk about climate change and all of the science around that next week. And next week, we're going to be talking about solutions to climate change.
That would be great.
We're going to have an interview with Jonathan Foley, Executive Director of Project Drawdown, next Wednesday.
Oh, I love Drawdown.
Yay. So it's very
Can I
exciting.
come into the show on that day?
Maybe. We'll
Can
talk
I join
about it.
in, please?
We'll talk about it.
Okay.
Okay. Say
I wanna
goodbye.
join. Bye.
Say goodnight, everybody.
Bye, Kai.
Night.
Nice to see you.
And that is the intro to the show today. Thank you for joining us. We'll see you next week. No. Thanks for joining us, Kai, to talk about the important issues that are concerning 8-year-olds these days.
Thank you.
I was worried about, like, I don't know what candy bar I was going to get at the store when I was his age. So things are a little different these
I was pretty worried about the rainforest when I was his age, but I didn't feel like I had any answers at all. It felt very so far away and done and over with and end of story and sad that I didn't really feel like there was anything I could do as an 8-year-old American.
Right. There are things we can all do. We'll talk about it. We'll talk about it next week. Should we do a show? It's time to do a show, isn't it? Let's do it.
Oh, is it that time?
Oh, it's time to do a show. I think we should have this thing that we've all come here for together today. I've got all these things open that I forgot to close the last time I was here. Oh my goodness. Oh my goodness. Hold on. Did I do what I needed to do? Goodness. Oh my goodness. As I check and make sure I have files before I delete things. Yes, I do. That's good. Quit. Save? No, just quit. Too many things open because I wasn't paying attention. I'm going to quit you too. I quit you, Firefox. There we go. And I got my iTunes because you got to have your iTunes. Doop, doop.
Bonk.
My bonk. You lose. Bum bum. Okay, starting the show in 3, 2. This is TWIS. This Week in Science, episode 717, recorded on April 17th, 2019. Keeping Science Alive. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your head with pig brains, fire ants, and irritable bowels. But first,
disclaimer. The following science news program is brought to you by people like you. More specifically, by people like Andy Groh. People who not only enjoy learning about science, but understand the importance of it to our everyday lives. People like Andy Groh. People who support science-based shows and science-based policymaking. People like Andy Grow. People who listen to the show and then tell other people about the show to spread the science knowledge to the world. People like Andy Grow. And while we have lots of choices to make when it comes to how, when, and where we spend our time in this life, we here at TWIS are always happy to spend that time here with you. And Andy Grow, here on This Week in Science, Andy Grow edition, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science that happened this week.
Yeah.
Yes, it's all in the name. It's what we do. So let's talk about it. What a great show we have planned for you. Tonight I have stories about printing hearts, pighead perfusion, and promising exoplanets.
Wow.
Yeah. What you got, Justin?
Oh my goodness. I have ancient pea archaeology, CBD for your head. Let's see. I've got irritable bowel syndrome. I've got some amazing environmental DNA test that's being done, and the universe's first baby molecule has been discovered.
Oh,
little
baby
molecule. Tiny little.
It's so cute. I just want to eat it up. Wait, I don't know if I want to do that. Yeah.
It'll be fine.
It'll be fine. All right, everyone. Or what's in Blair's Animal Corner?
That's all right. I'll just go. I have fire ants, I have sea spiders, and I have primordial ooze.
Oh, that's the good kind.
Blurp. All right. As we jump into the show and await the primordial ooze to perfuse our brains, I would like to remind you that if you have not yet subscribed to This Week in Science, it's pretty easy to do. If you're interested, you can find information at twis.org. We are also available all places that good podcasts are found, as well as YouTube and Facebook. So, on to the news. Okay, what is in the big news rundown this week? 3D printing of organs. This is the gold standard of, oh my goodness, someday we are going to print the organs that we need. We are going to not have an organ donor shortage anymore. Everyone will have organs when they need them. And so challenges have been set to bioprint aspects of organs. Some organs are easier than others, but one of the main problems is the perfusion of blood through the tissues to enable all of the tissues to come together to function as a whole and to have the nutrients they need so that the organ, all the cells in it, can survive. Another aspect of this progress forward is not just printing organs, but potentially using people's own cells. So taking biopsies, samples of people's own cells, and then growing them into these organs. Right? Very exciting idea. And this past week, researchers published for the first time in Advanced Science, a team out of Tel Aviv University, the very first 3D printed heart, which you would think this is a huge deal. So they took a biopsy of a patient's fat cells. There's a fold of fat cells called the omentum in the abdomen that wraps your organs, and it's fatty, and there's wonderful extracellular matrix and also cells in that area that can be used for experiments such as this. They took the cells themselves and got them to regenerate, and they started creating populations of this patient's cells, and they told these fat cells to turn into heart cells. And initially, they and on top of doing the heart cells, they decided that, oh, hey, we will move on from just heart cells. We will use this extracellular matrix and combine it with some other compounds that we have and create a biofilm, basically a biological matrix to grow the cells in. So, with experiments that have led up to this point, this kind of growth medium has very often come from animal cells. You find a medium that can support the growth of cellular structures, and it very often is not from the patients themselves. And so they have developed this patient-specific biological matrix, so they can print the cells. They can also grow the cells, and they can support the cells all from the patient's original tissue, which is a huge step forward. Initially, they were printing heart patches, so using 3D bioprinters to just print little patches that had the shapes of which had come from scans of the patient's heart. And they were like, oh, we can fit this over this little vessel here. We can fit this patch over this place here. And they showed that they could print these patches with vascularization and with all the stuff, and they could support the growth of these small patches that could fit on a patient's heart. And then they were like, well, let's just see what we can do. And they scanned the patient's heart, and then they're like, let's print it. And they did. Key problems here, though, is that it's the size of a rabbit heart. So it could fit in the palm of your hand. It's not quite big enough to actually replace your heart. This
Just
wasn't
a tad smaller than I would like.
Yeah,
in
not
my
a full size printing. So we're still dealing with the limitations of being able to make sure that we can really grow tissue the size that's required for the human body. We're still dealing with that. But they have this little tiny rabbit heart-sized printed human heart that seems to have cellular contractile activity. However, it doesn't play well together. So the cells aren't really contracting as a team. And so that is the next step, to get these cells starting to work together so that this kind of printed heart can actually function. Maybe not in a person, but could be shown to really have atrioventricular contraction and function to pump blood. And then potentially someday down the road, we're getting there. We are honestly starting to get to the point of printing organs. This is a big step forward. And I just kept laughing at all of these headlines. This first 3D engineered vascularized human heart has been printed. And I was like, yeah, that's amazing. I didn't know we were that far along in the technology. And then I looked and I'm like, oh, but we kind of didn't.
Yeah, it's very close. I feel like we're getting so close, though. My question is going to be, I mean, obviously the huge benefit to this is that you don't have to wait on a transplant list. You don't have to wait for something that is compatible. You don't have to bleach out other people's cells first. There's all these barriers that are removed. The question is, once we figure out how to do this, how expensive is it going to be? Because is this something that only a very select number of people are going to be able to do? Or is this the exact opposite? Is this something that once we break through that wall of technology, is it actually going to be more accessible than a transplant? I'm just very curious.
Yeah. Well, I remember we saw a 3D printer when we were in Philadelphia, and though these 3D printers are based on off-the-shelf technology. Some of them are much more advanced, but many of them are based on off-the-shelf technology, and they are not incredibly expensive devices to create. It is the processes and the procedures that really are the stumbling block. And It's interesting, though, that the way that they're doing it is a fairly efficient process where they have the cells growing as they're doing another step. And then they kind of, so everything is all the steps are overlaid. So it makes it a faster process. Um, but initially, this kind of technology will always be a very expensive
Yeah.
thing to do. This
I
will be a
guess
very expensive endeavor.
what
Yeah.
I'm really wondering, which this is the cart way before the horse,
Yeah.
but I'm kind of wondering who is going to hold this technology and who's going to be in charge of dispensing 3D printed hearts. Is it something that will live
Mhm.
inside a hospital and we will be part of a copay? Or is it something that will become some sort of prescription? You'll have to get a prescription 3D printed heart from a
Right.
pharmaceutical company. These are kind of what I'm wondering. This is what I'm wondering is once this becomes a production item, who holds it? It's kind of this undiscovered territory in the medical field that I'm very curious to see where it goes.
Or it could end up in the hands of a democratized scientific, I think, where everybody has their own printed heart that they've already pre-printed.
Yes,
And they've
ready
got
to
in
go.
their organ closet at home,
Yeah.
just in case.
Just in case.
Depending
Yeah.
how far ahead you go.
And there is a lot in the technology, especially out of universities, these technology fields, where people are creating things fairly open source, which is provocative in that sense. And the researcher who is behind this study says we need to develop the printed heart further. The cells need to form a pumping ability. They can currently contract, but we need them to work together. Our hope is that we will succeed and prove our method's efficacy and usefulness. Maybe in 10 years, there will be organ printers in the finest hospitals around the world, and these procedures will be conducted routinely.
Okay, my ticker's just got to hold out 10 years.
I don't know about 10 years, but maybe. We'll see. I don't know. I love the way it's moving forward. And speaking of technology moving forward and ability to do things, it's just mind-blowing, this next story that I brought. You may have seen it in the news. Researchers have published in Nature about their Brain X system. It's a perfusion system that has enabled them to the sensational spin on this is bring pig heads back from the dead.
Is this a head in a jar? Is that what this is about?
It's not a head in a jar, but it potentially could be a head in a jar
What?
if taken the wrong way. So let me tell you about this. What they have done. This was reported on about a year ago by MIT Technology, and the researchers behind this process have been working on it for quite some time. They, in this particular paper, took pig heads from an animal facility that these pigs had been sacrificed for food, and they took the heads. And 4 hours later, they split the group up and they had their tests and their controls. The controls got not their Brain X perfusion system, but or they got perfused with stuff, but not the Brain X solution. However, the test group, they used their Brain X system, which allowed the restoration and maintenance of microcirculation and molecular and cellular functions of this intact pig brain up to 4 hours post-mortem. So they took these pig heads and they plugged in the artery in the jugular vein, right? The carotid artery jugular. They perfused the pig heads with this solution of nutrients, and also it also contains ion channel blockers, which is important for a point that I'll bring up in just a moment. This what they call extracorporeal pulsatile perfusion system, which is out of the body and it pulses like a heartbeat. So it pushes the perfusion fluid through the brain so that even after this animal has been dead and had no blood, no nutrients, no oxygen going to the cells of the brain, we know that within minutes after death, after the heart stops, the brain starts to shut down because it has no oxygen. After an hour, maybe up to 2 hours, you start to have cellular apoptosis because the cells themselves, the pH increases, and the cells start going into metabolic shutdown. and killing themselves, and they start bursting, and a liquefaction process ends up happening. But there's this whole process of death that we know that takes place. Now, not all the cells die all at once, right? So there's something that's happening with this perfusion system with the nutrients that they have included. It's a hemoglobin based, non-cellular, non-coagulative, cytoprotective solution that they've put in there, and it allows recovery from the lack of oxygen. It also, when other experiments have done in the past that have tried to do this kind of reperfusion and bringing back oxygen into the system, there's usually injury to the system during that process, and what they've built doesn't injure the quote unquote dead head any further. It prevents swelling and edema, and it metabolically supports the energy requirements of the brain. And so when they looked at EEGs, there was no brain activity per se, but there was cell metabolism. There was a decrease in cell death. There was preservation of the whole structure, the cytoarchitecture of the brain, as opposed to it collapsing on itself through that decomposition process. Restoration of vascular and glial inflammatory responses. There was also there was spontaneous synaptic activity, but not enough to create a collective, what you would call a brain activity. And there was this metabolism that took place. So the brain was brought back to life. It was resuscitated in a sense.
Until it responds to stimuli, I'm not there.
I am. I don't need an outside world. That's why I have the do not unplug order standing, even if I'm in a comatose state. That what you're calling sporadic activity may be all I need.
So part of this, there is a huge bioethical aspect to this. You know, if we can potentially resuscitate a brain, what does that mean for brain death? At what point is a brain truly dead if hours after supposed death, a brain can be resuscitated? Now, in this perfusion system, I mentioned that there are ion channel blockers, and that is important because it decreases the ability of the neurons to fire, and that is part of the system. They don't want necessarily the neurons to fire in their normal activity. They did not want to bring a severed pig head back
Right.
to life and have it conscious
Right.
and
I'd
aware
imagine
and
there's some
feeling pain.
Yeah.
Yeah. And we don't know
Yeah,
what
ethically,
we don't know what
that's
they
rough.
would feel.
Yeah.
And so the ion blockers would reduce neuronal activity, and they did not see any EEG as a result. But people are still kind of going, well, what does that mean? And what would happen if you removed the channel blockers, the ion blockers, and perfused the system with this resuscitative juice, right?
Just at least once. Like,
What would
see
happen?
what happens.
Yeah, I'm all for it. There's gonna be some anger guaranteed when that happens.
For sure.
But if this leads to head in a jar technology a la Futurama,
Yeah.
I'm for it.
Can I
think it's great.
sign up? Can I sign up for the if I should die test where they would try this out on a
Oh,
human
yeah.
cadaver? Like I would donate my brain to this study.
Right. I donate my brain to science if it goes to this study.
Yeah.
Exactly.
Yes.
Yeah.
I know it doesn't work that way, but we know the brain guy. Maybe I could talk to him. Be like, hey, I know you know where to take my head. If anything bad happens, let's get this on paper.
So the question though, so where is this going to lead? The researchers are developing this technology in part, there's multiple reasons, but in part because of the lack of ability to study intact brains after death. Like you can't study a human brain easily in someone's head while they're alive. You can't study a human brain even after death without perfusing it with stuff that sticks it in a static state. Right? When you put a brain in formaldehyde, it stops. everything and freezes it in a particular state. And this potential system could allow researchers to do brain research after death or in brains that have taken from animals that were involved in a research study, and they want to look at how things connect within the brain. They'll be able to do this in a brain that is not decomposing and is still cellularly, metabolically active without the potential ethical issues of it's a living animal. Even though people are raising the question of, well, is it a living brain
Right.
at that point? The second aspect is this could someday be used as maybe recovery for stroke. If somebody is caught with a stroke or some kind of terrible brain injury, could this technique, this Brain X solution, be used instead of the current medical techniques and treatments that are used to help people get through that kind of stuff? And then finally, yeah, brain in a jar. I'm wondering, if it's if this works this for the brain, are people going to want to put their whole bodies in this solution and say, okay, preserve me forever. Here I am. There we go. Forget cryoprotection, right?
Yeah.
Forget
Just-
cryopreserving. We don't need to freeze it anymore. You just get to be in this.
Go for a nice swim.
Perfused.
Yeah,
Mhm.
one
Go for a
power
swim.
outage, and then you're, in a non-sufficient battery backup, and the cryo fails.
You gotta have a battery backup.
Yeah, but nobody ever puts enough of a battery backup there.
I mean, hospitals are pretty good at that. That's kind of their job.
Yeah.
Battery backups? I would hope
Generators.
so.
Yeah.
Hmm.
There you go. We figured it out. That's how I'm gonna live to be forever years old.
This is how you will
Forever.
be forever young. That's right. This is This Week in Science. Justin, what did you bring?
I brought Cannabidiol, aka CBD, which is the non-psychoactive, or one of the non-psychoactive out of the many compounds that can be found in cannabis. And we have talked about a bit about this in the past. The many potential and found benefits, ranging things from there's potential cancer, maybe even not to the point of the cures, but what do you call it? Versions, concussion protocol treatments. There are usages for helping people get off worse drugs like opioids. They actually have some pain relief, epilepsy. All these things are known to be in here. Scientists have now shown CBD could be used in a completely different way, one that they hadn't envisioned before. They can use it as the Trojan horse to slip medications past the blood-brain barrier. This is an ACS journal, Molecular Pharmaceutics. The blood-brain barrier is a sock that's around your brain. What is the blood-brain barrier, Kiki?
The blood-brain barrier are just the very tight junctions between cells in the tissues that protect the brain. So instead of having open channels where lots of stuff can flow back and forth like we potentially have in our guts, you have a very tight barrier where it's highly controlled so that things that shouldn't be there don't get in.
So some of the things that get in, there's some amino acids that are let in. It's a very exclusive club. Glucose is allowed in. Neurotransmitters, is how would they be transmitting if they couldn't get past the blood-brain barrier? So there's an entire class of neurotransmitter called endocannabinoids. These bind to proteins called cannabinoid receptors, and this is also something which resembles very much what CBD looks like, which is why it's cannabidiol. This is how these connections, we had the molecule and then we found it in the brain, or vice versa, very similar. So what they did was they created a little pocket, a lipid pocket onto the CBD. And as proof of concept, they put a fluorescent molecule in that pocket. And they showed that the CBD managed to get past the blood-brain barrier. Now, they had similarly sized pockets that didn't make it in as often. So when injected into healthy mice in this research, CBD with the glow sticks in their pockets transported about 2.5 times more fluorescent molecules to the animal's brain. Which means like it's a very high potential that this is going to be a good method for getting drugs, that sort of thing, past the blood-brain barrier if needed. So maybe a nice delivery mechanism.
Yeah, and there have been other delivery mechanisms we've used, like viruses and things that are, they're small, and so they fit through those tight junctions. And wow, viruses are potentially dangerous. So that's been one of the big issues. And so stuff like Cannabidiol, which we know is it's something that people use constantly. It interacts with our endocannabinoid system. It is not, it could be modified to not be active in certain sense, but to carry drugs that would be active in another sense, and that would be amazing.
Yeah, and CBD in your brain isn't doing anything terrible.
No, it's not.
Let's see. Oh, this is interesting too. This is a correlation of sorts, but one that you would kind of expect to find. So I'm not sure how. But so far, this is just a small sample size of 23 pre-adolescent children with irritable bowel syndrome and 22 healthy controls. Participants were asked to maintain daily pain and stool diaries for 2 weeks and to provide some samples. And what they did was they did a bacterial composition of their microbiome. And after some use of metagenomics, creating some metabolic markers, they were able to distinguish just from samples those who had irritable bowel syndrome and those who did not, with 80% or greater accuracy, which is still a little lower than I might have expected. But I'm actually surprised that this isn't a thing that we've already really delved into. But they had this classifier system. Classifier assesses specific metabolites, types of bacteria, functional pathways, other factors, and they can sort of now see, like looking at the microbiome, pretty good chance that you do or do not have irritable bowel syndrome. Which the fact that they had 80% or greater accuracy in distinguishing is actually not too bad, seeing as only about 20% of the population have it. 50% of their population in this study had it. But yeah, so there is they're concluding that there is definitely at least a very strong correlation between your gut microbiome and whether or not you have irritable bowel syndrome.
Yeah, that seems like a slam dunk. It seems like it's. I'll tell you, as somebody who in my workplace they call it, I have TPS, tissue paper stomach. I'm somebody who can get stomach aches like that. I've often thought about, God, I wish I could just take a poo pill, reestablish, have a little bit more of a lead gut. I'm so sensitive.
Right. So what's really great about studies like this is once you identify what those microbes are, once you identify perhaps what the because they have some of them that were shown to be having that were increased when there was the unhealthy affects, and some that were a little bit increased with the healthier patients. When you kind of have that, you no longer just have you can actually just raise those microbes up and take a pill that was never poo at any time. So this is a novel approach, but that there is another way.
I'll take it.
Just probiotic, but very specific probiotic.
Very specific probiotic.
I feel like it's only a matter of time until when you give blood, you are also giving a stool sample so that you can have a baseline microbiome in your medical file.
Oh yeah.
I feel like we're very close to this.
Yeah. And the other
I would prefer a cheek swab if possible.
That could work too, but then you can't really tell what's in the lower intestine.
No, it's probably if 33% didn't make it, or 66% didn't make it through. So the other part of it though, then I was it's like you could actually create a bacterial phage. You could like if you identify the bad players, you could potentially engineer something that would go in and devour only them. That was specifically designed to go after bacteria that cause or increase the symptoms, the inflammations of irritable bowel syndrome. And so the pill you take might not be a probiotic, but might be a viral phage that does the same job. There's going to be a lot of ways to address and attack this. It's going to be very interesting.
But I think what I think is interesting here, though, like you mentioned the 80% number of being able to identify whether it's a healthy control or someone with IBS. I mean, that still leaves a very wide swath of potential false positives. And in that case, you know, when you're trying to put someone toward the appropriate treatment, that's still I mean, you're closer than you're not, but it's still a kind of
this is correlation level in investigation at this point.
Yeah.
I still feel like this is just the start to going, okay,
Right.
this is the bacteria we find in people with IBS very often. Right. Let's remove that bacteria. Oh, your IBS went away? Great. This is what we need to target for IBS cases. So like, I still feel like we're 2 or 3 steps away from the solution here, but I think it's an important establishing step, right? Is to isolate the bacteria that IBS cases have in common.
Yep.
And also, and like you said, this is a sort of, we talk about correlatives as though they're not sufficient. Correlatives are great places to start. You know, correlatives are the thing that says, hey, I found this correlation. Now fund the next round of research, and let's go with a 500 person survey, and let's go with deeper sequencing and see if we can find more depth to the microbiome.
Then let's see if I can give someone IBS.
Yeah, no,
that's part of it.
That would have to be a thing.
Yeah. Oh dear. No, I don't want to do that to anyone right now. We're working on your brains.
Yes.
This week in science. Is it time for Blair's Animal Corner?
Oh, probably is.
All right, it is time. Yeah,
let's do it.
Blair?
Loves are creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Oh, I have a story as old as time about fire ant colonies. You know, you have your single queen. She's in charge of all that she surveys. But then over there across the road, there's a colony with many queens. What do they have going on over there? Well, this is a study from University of Georgia looking at tropical fire ants native to Florida and coastal Georgia, looking specifically at colonies that thrive with multiple queens in close proximity to single queen colonies of the same species. So this is why the lead researcher, Kip Lacey, was so interested in this, was because they were right next to each other, but they had these completely different social structures. This was looking at these tropical fire ants. They isolated communities of native fire ants that cover the shoulder and medians of Florida highways. So that's another reason that Florida, maybe not the place where I would like to reside, just fire ants on the highway. It seems really intense.
That's the name of my next album.
Fire Ants on the Highway?
Yeah.
Yeah. Is that a metal record? I feel like it's metal, right? So they found that in these areas, the multi-queen, and I always say this word wrong, polygamy colonies, yes, would be nestled right next to the single queen monogamy colonies of the same species. Nests with single queens were as close as 5 feet away from nests with as many as 13 queens. Many fire ant colonies were founded by a single queen mated to a male from a different colony, so that way they have the unrelated genes, right, who produce all of the eggs that start a colony of their own. In polygynous colonies of this species, all the ants shared a social chromosome. So they found that the reason these guys were did have this very different social structure was down to a specific chromosome that was absent from individuals in single queen colonies. This is what they dubbed the polygyny syndrome. They also found that this chromosome led to lower levels of aggressiveness towards non-nestmates and smaller body sizes of individual ants. Where this gets interesting is that there are other types of ants that have these two different social strategies, but there is something that is totally different here. So in imported fire ants, monogamous colonies produce all their daughters sexually. But in contrast, this tropical fire ant colony in Florida with multiple queens found that the queens reproduce asexually.
Huh.
So reproductive queens and daughter queens inside a colony were genetically identical, but the worker ants in these colonies displayed greater genetic diversity. Unlike the queens, they were not clones. When they did some controlled experiments with these ants, they found that the queens were produced asexually and workers were produced sexually in polygynous colonies. They also found that the male ants in these colonies seem to be sterile. Okay, so let's unpack this. So we have a bunch of queens. They are reproducing asexually to make daughters. They are reproducing sexually to make sons, and those sons are sterile.
So where's the
queens from multi-queen colonies were mated to males from the single queen colonies next door.
So they were symbiotic, even though they had completely different strategies.
Here is the weird thing. I would not call it symbiotic because this helped the multi-queen colonies by introducing new genetic material. But because those males were sterile, those sons, there is no benefit to the male ants from the other colonies. So they are deeming this sperm parasitism. Yes, sperm parasitism.
So taking advantage of the diversity, but then not taking advantage of that for future generations.
Correct.
Yeah.
Because she's got to take care of herself, make her genetically identical daughters to make sure, it all stays. So this is just insane. This is something we haven't really seen before. Why this is important ecologically to learn this kind of stuff is that, first of all, you have these invasive fire ants in Florida that are different from these fire ants that were in the study. So we need to know how the social structure and the genetic kind of descendants works in the local species so we can protect it. But additionally, there are invasive fire ants in coastal Georgia and Florida as well. So if we can extrapolate information on invasives from this study and find similar systems, it may be easier to control those species. But mostly, I just think it's very interesting that there are these two different chromosomes that create two different social strategies, but these two different social strategies are somehow still intertwined, but against one half's will.
Right. The females that are producing fertile offspring, the female that is going and having her diverse non-clone army of fertile offspring, that female she's like, this is my thing, it's great, and the other female's like, no, I'm going to take advantage of that right there.
Yeah, but you're just talking about this from the perspective of the females.
Right.
The male ants that go and visit the queens on the other side are like, it's actually a pretty good deal.
Actually, remember, Justin, outside of our species, sex is supposed to be for reproduction. So the hormone drive to mate comes from a desire to perpetuate DNA.
Sure.
So but if you're making sterile sons, there's no evolutionary driver to mate with those females.
But can I point something out? The visiting male ants, they don't know that.
That's why it's
They don't know
parasitism.
that.
They're tricking them.
Yeah. Yeah, you know what? They don't
Trixie.
Yeah.
care.
Yeah. Well, anyway,
They're fine with it.
it's tough to be a fire ant.
I'm an ant, but yeah, tough enough to be a fire ant. What am I going to worry about all that?
Yeah, well, there you go. From one creepy crawly to another, what do you know about sea spiders?
That
Spiders
they
of the
are
sea.
in the sea and that there's yeah, the
Yeah.
same as Kiki.
They're bizarre looking. Everyone listening who is not watching the show right now,
Whoa, I just
please do a quick Google search for sea spiders. You can also check out my Twitter, my Facebook for a GIF of them walking. They are fascinating. They look kind of like crabs missing bodies. They're just kind of a lump of legs, but they are in fact related to spiders. And sea spiders are famous for getting enormous because they live in very, very cold waters. And this has been a long believed theory that there are animals that get huge in deep ocean waters because this is called the oxygen temperature hypothesis. Animals living in extreme cold can grow to giant sizes because their metabolisms are slow and therefore they don't require as much oxygen. It takes a lot of energy to move oxygen through a larger body. And so by being in a colder temperature where their metabolism is slower, they need less oxygen, they can grow bigger. This has been the theory for a very long
for
time.
a spider?
We'll see if Kiki can pull up some pictures, but
Mhm.
they
I'm
get
working on it.
huge. They get as big as me.
What?
Yeah, they get really
What?
big.
No. What?
Yeah, they get. I've seen
Now,
some
to
in.
be fair, for those of you who haven't met Blair in person, she is only 3.5 feet tall, so it's not as big as
Nope.
you might think.
Yeah.
Wait, what?
They can get not quite as big as me. They can get to be about 3 feet.
Okay,
So
yeah, so 6 inches shorter than you.
Yeah. So I thought they could get a lot bigger. I thought they could get to be 5 to 6 feet, and maybe they can. Maybe I found a bunk source, but they can get to be really, really big. Which for a spider, if you think about spiders, remember they're
Oh no,
spiders.
that's massive for a spider.
Huge.
Yeah.
So that was the theory previously. This is from University of Hawaii. They did some research looking at how metabolism is related to body size and oxygen distribution. So before I get any further, let me also tell you that sea spiders are quote unquote skin breathers. They do not have gills. They do not have lungs. They rely on diffusion across surfaces on their legs.
Oh, their
This
legs
looks
are
like-
their lungs. They have lung legs.
Lung
Wow.
legs.
This
Lung
looks like the what is it? The
Daddy long legs.
Orson Welles play where it's the end of the world and the aliens are attacking.
I don't remember there being sea spiders in that.
Yeah.
Anyway, in this study, I love this. They quote unquote exercised the spiders to exhaustion. How do they do that? Flipping them upside down and counting the number of times they were able to right themselves. They did this at a range of temperatures from their
That would
normal
be tiring. Yeah.
Yeah. Their normal temperature of negative 1.8 degrees Celsius all the way up to 9 degrees Celsius. Counter to predictions, these sea spiders kept up with smaller ones at every temperature. Despite their body size, they were able to right themselves consistently. So, I mean, I just love the idea, first of all, of like an animal getting up and then you pushing it back over and it getting up, pushing it back over.
And this is
Getting
science.
Wait, and you work
Pushing
at a
it back
zoo? You work at a zoo.
I
Is this
think
what
it-
happens at the zoo after hours?
No.
People are out there pushing
However,
over zebras.
I think it's very funny
Tripping
that
the
some
ostrich.
researchers got together and thought, okay, how do we exhaust a sea spider? Oh, I know. Just push it over. Then it gets up, push it over. Anyway, so counter prediction to predictions, they kept up with the smaller spiders. They dealt with warmer temperatures just like smaller spiders, which means that the this is not the reason for them to be able to grow so big. It has nothing to do with the water temperature, at least in this very small preliminary study. They used microscopes to look closely at their legs. How the heck are they getting this oxygen from their legs throughout their body in higher temperatures? They found that sea spiders are covered in pores. Quote, the exoskeletons of these really big ones look almost like Swiss cheese.
What?
As the spiders grow, their exoskeletons become more and more porous. So they allow for better diffusion of oxygen across their legs as they get larger because there are more and more holes. Again, the researchers caution this is a short-term experiment. A long-term effect of warming on giant animals are not yet understood. But at this time, it looks like these particular giant animals may not be as vulnerable to ocean warming as we thought. And
Yay,
the
the
previous
future will be full of giant
hypothesis,
spiders.
yeah, is not the oxygen temperature hypothesis of large deep sea animals may be bunk.
I think it's really fun when research finds evidence against
Best
hypotheses.
Yeah,
Right?
it just means you have to go back looking for more.
What is the explanation? I mean, maybe it's that they live in water and they don't have and because they don't have to deal with gravity so much because they're surrounded by the liquid water to loft them. They just have to deal with pressure.
It wouldn't,
No.
if they're deep sea, wouldn't
But
gravity
it's
be
pressure.
worse?
Well, it's gravity, but there's the water that's,
Mm.
around them and kind of shielding them because they float in the water.
Right.
But density and pressure is a huge issue in the deep sea. So they're still dealing with massive amounts of pressure. So that could be an issue,
Yeah.
which you wouldn't expect things to get giant under high pressure, and they do.
I guess once you're built to withstand it, and it looks like a pretty, strongly put together, thin limbed has got to be an asset. You don't get to bulk up too easily, structurally under that kind of pressure.
Right. Okay, so also I found out that the reason I thought sea spiders got as big as me, this is very funny, is because there's an animal called the Japanese spider crab.
Oh, that
Which
gets huge.
does get as big as me. But sea spiders still get to be about the size of a trash can lid, which is very large.
Big enough for a spider.
Big enough.
Oh my goodness. And on that note, spiders of the sea, we are going to head to a break. We are going to take a short break and come back in a few moments with more This Week in Science. We have some space news. We've also got some baby molecules coming up with more This Week in Science. Stay tuned.
Can't explain things you've heard more than intuition. A line of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scatter plot of first assumptions were correct. Disprove the rest. It's hot. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't.
Thank you for listening to this episode of This Week in Science. We are so glad that you are here today with us. If you enjoy this show, please consider supporting us and helping us maintain our listener-supported status. There are many ways that you can support This Week in Science, so let me tell you about them. One way is to head to TWIS.org, where you can find all of the links for various ways that you can support us. That's where you can find everything is at TWIS.org. So head over to TWIS.org And once you're there, you can click on the Zazzle store link at the top of the page. That will take you to our Zazzle page, which is full of items that have the TWIS logo, polo shirts with a nice TWIS icon on it. We have pillows and mouse pads and other things that are covered with art from previous Blair's Animal Corner calendars. And every item that you purchase, a portion of the proceeds from this Zazzle store go to support This Week in Science and help us keep doing the things that we do here every week. Also, you can, from TWIS.org, click on the Patreon link. Patreon is a crowdfunding solution for shows like ours. For creators like us, we use it to be able to support what we do through donations. People support us as patrons, like a patron of the art. You can go to patreon.com/thisweekinscience or click the Patreon link at twist.org. On the page, there is a big red button that says Become a Patron. Click on that, and it will take you to our Patreon page of donating selections where you can choose the level of your support. You will be charged once per month at that amount, and you will receive fun gifts in return for the various levels. $10 a month and above, we will thank you by name on the end of the show. At the end of the show, we run off a list of names if you've heard that before. Those are all people who support us at the $10 a month level or above. Don't you want to have your name read? Want people to know that you support TWIS? That's right. Be part of our patron team. Be one of our patrons. Back at twist.org, also, one of the great ways that you can support us is to subscribe or get other people to subscribe. You can tell people to go to twist.org, click on that subscribe link. It'll open up our YouTube, iTunes, or Google Play options. Those go directly to their subscription options at those various platforms, allowing you to subscribe and get TWIS each and every time we upload an episode. Everyone out there, if you're subscribing, we thank you so much for subscribing to this show, for bringing us into your life every week. Are we in your ears while you're on your commute right now? Thank you for sharing it with us. Thank you for being a part of TWIS. Thank you for supporting TWIS. We really could not do this without you.
Can't explain the things you've heard with more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere.
And we're back to This Week in Science.
That's right. We are back with more This Week in Science. And right now it is time for the part in the show where you share with us. It's time for This Week in What Has Science Done For Me Lately?
Lately, slight delay
And
over here. Sorry.
little delayed. It's all right. This week, Tedward writes in and says, So, my doctor recently said that I needed to talk to a cardiologist. He said that I needed to get an angiogram to find out if I had any heart blockages. I was in and out within 8 hours and went to work the next day. The results of the angio said that we probably need to go in and do a triple bypass. That's a big deal. So we're setting up everything with my insurance company to take care of the triple bypass. I work part-time at minimum wage, and I absolutely do not have a company health plan. And while medical science is doing all the heavy lifting, there's another kind of science that's paying for it. Thanks to the Affordable Healthcare Plan, I have medical insurance for one of the few times in my life. Thanks to the state of California, I have Medi-Cal because my income isn't sufficient to warrant me paying for insurance. And say what you like about it, I find the coverage is vastly superior to every other medical plan I've ever had. So, what has political science done for me lately? In the stroke of a pen, I'll be able to get my heart repaired and recover and even work casually because of medical science and have it all paid for through the taxes I've been putting in all my life. Tedward.
So
Nice.
fantastic point. All of the efforts that are made are nothing without a little bit of political policy to back them up.
Yeah. And this is
Very fantastic. What has science done for me lately? I love it.
Yeah, I think it's a wonderful spin on our weekly segment, and I was very happy to read it. And I hope that the surgery goes well and that you do recover well and are able to continue to work casually after, and that everything go. I just really hope everything goes great. It's a combination of the medical science, the science that backs it all up, economics, which is also a science, political science, all these things working in tandem.
Nice.
Don't we all deserve a little bit of health? And
We
everyone,
do.
remember, remember that we need you to keep writing in and sharing in this segment of the show. This part of the show is all from you, and we really love having it on the show. Message me on Facebook, our page, facebook.com/thisweekinscience, or you can email me at kirsten@thisweekinscience.com. Keep it up, everyone. Help us entertain you with your stories. Justin, what'd you bring?
So I actually brought a story from Hawaii at Manoa Department of Biology, which I think Hawaii is also where the spider story.
Yeah. Yeah. They're
Sea
busy.
spiders.
They're
Yeah.
busy this week.
So they've developed a technique for measuring the amount of living coral on a reef by analyzing DNA in small samples of water, of the seawater. This is research by Patrick Nichols, graduate student marine biology graduate program, and Peter Marco, associate professor in the Department of Biology. And they have published in Environmental DNA. So a while back, it was in early March, we talked to Raquel Pesciuto, whose name I'm probably mispronouncing. I'm sorry, Raquel. Episode 711. We talked to her about some of the difficulties involved and some of the successes in studying microbiomes in these environments of getting in there and getting the DNA of that which is swooshing around. So this analysis, of course, didn't have to go as in depth, but what they were doing is environmental DNA. So they take water samples, they sequence everything in there. They're looking for genetic residue that's in there. They sequence it, and they take those sequences and they match them to references of known corals. And by doing this, it turns out, voila, they got a snapshot of the health and diversity of the coral down below. Without scuba diving in, without submarines, without a really prolonged study.
Just a little ladle into the water.
Yeah, that's kind of how it's presented here. So the technique takes advantage of the fact that the coral shed like any living organism. They're constantly dropping bits of their DNA out into the environment.
Wow, that would be like going into like the dustbin or the little dust scoop at a museum. After somebody's done sweeping up after a day at the museum, what does it hold? Who was there? Yeah.
So there actually have been this technique in form has been used to look for ancient hominins by sampling DNA fragments found in dirt layers. So there has been the environmental DNA has got a huge potential, and it's just getting tapped into. And this is one way. But so what it's sort of, and they did also do some testing on dying reefs, and they could see that there was this significant drop in both the diversity and the amount of DNA. There's some level of sort of figuring out if there's corals that put out more, that shed more than others. But they were actually really able to get a decent snapshot that they could compare at later time points. Also, what was sort of interesting is opposed to going down with divers or even a little submarine and taking pictures and visually inspecting, they were able to pick up rare coral that might get missed in a visual inspection. because because they're hidden. Yeah, they're hidden, or there's just less of them, and you
Yeah.
They're
don't
like,
get
I'm in
maybe.
here between the big corals.
But they were visible. They were visible through this technique.
Yeah. Yeah. So I remember, gosh, it was probably years ago. I did a story about environmental DNA and following fish migration with it.
Mhm.
And I remember that just feeling so weird that a moving animal just through the water column and something as big as the ocean.
Right. Yeah. You'd think
That
things
you
are
could
just
just
it's in solution. Everything's
pull
everywhere.
some water. There it is. But yeah, especially in a coral reef, in some of the most biodiverse spaces on the planet, with so many plants and animals occupying that exact space. And then also, let me just remind everybody, we share about 60% of our DNA with a banana. So that also means there's a lot of DNA that's kind of overlapping. So
Yeah.
you have to really be good about knowing what coral sequences are, and then being able to find that. It's something that, even 20 years ago would have been totally out of the realm of possibility for us. But because DNA sequencing has become a lot easier, it's something that's much more within reach. And I absolutely love this idea. There's so much of the ocean we know so little about, but just scooping some water, we can figure out who's living in the neighborhood.
Yes.
Yeah. And even just scooping some water, there have been some huge projects mapping the microbiome of the ocean, mapping what is out there and getting what reference signatures we didn't used to have. So now we have more signatures of life that is out there, and that involves more coral, more fish, the microbiome. Our information is growing, and that is only going to enable us to do more. Yeah. Speaking of places that life could be, what about exoplanets?
Oh,
Yeah.
that's a good place to look.
Yeah, if it's going to be somewhere.
Let's look at
Yeah.
exoplanets. But one of the big questions, a lot of these exoplanets that we're finding are either very close to their host stars, or they are around stars, red dwarfs, that tend to have lots of frequent solar flares. And solar flares means radiation. And if there's a lot of radiation ramming through your atmosphere, do you have an atmosphere? Is the planet protective in any way? Or is it just the radiation running amok as it will? Some researchers from Cornell University, astronomers, have written a paper and published it in the April 9th monthly notices of the Royal Astronomical Society. Lisa Kaltenegger and Jack O'Malley James have written lessons from early Earth. UV surface radiation should not limit the habitability of active M star systems.
Consequences. Consequences are taking place.
So they looked at planets near Earth, so that are also around these type M, active M stars, which are red, like I said, red dwarfs that have these frequent solar flares. Proxima b is one of those stars, only 4.4 light years away. It receives 250 times more X-ray radiation than our planet.
Oh, that can't be healthy.
Right, can't be healthy, right?
Unless you evolved there.
Exactly. And the other side of it is that at one point in time, Earth was bombarded by a lot more radiation and didn't have as thick of an atmosphere, although it did have a magnetosphere. And life evolved here. So maybe the other side of it is that radiation is important for evolution. And so they simulated, they modeled the surface UV environments of TRAPPIST-1e, we know the TRAPPIST system, Proximab, another planet, Ross 128b, and LHS 1140b. And according to their approximations, that even though we don't know what the surfaces of those planets are like, it's really highly likely that even with a significant amount of radiation, life would still be able to exist on those planets.
Nice.
Yeah. Yeah, that is nice. And so then they also looked at an extremophile here on Earth, Deinococcus radiodurans, and it's one of the most radiation-resistant organisms that is known to man. And so they were trying to figure out
Could it survive on one of these planets?
Yeah, they were looking at the mortality rates at different UV wavelengths of this extremophile, and they wrote a dosage of UV radiation at 360 nanometers would need to be 3 orders of magnitude higher than a dosage of radiation at 260 nanometers to produce similar mortality rates in a population of this organism. So basically, these different frequencies need different dosages to create death. However, it would be at 360 nanometers. If that kind of UV light is hitting the surface of the planet, there would have to be a lot more of it to kill off this radiation resistant bacteria. So what they were looking at, they're really showing that UV radiation should not be a limiting factor for the habitability of planets orbiting M stars. Our closest neighboring worlds remain intriguing targets for the search for life beyond our solar system.
And we now know we have an emissary that we can send ahead.
That's right. Send the Deinococcus. Send the radiation-resistant bacteria.
Yeah.
And speaking of exoplanets, NASA's TESS mission, which is the follower to the Kepler mission, is looking for exoplanets, and it has found its first Earth-sized alien world, HD 21749 B and C. So there's one that's an Earth-sized planet, and the problem with it, however, is it's HD 21749C. It probably doesn't have a potential for life existing on it, seeing as it's very close to a very hot star. The star 21749 is almost as big as our sun, and this planet goes around it once every 7.8 days, which means that it's very close. So it's probably burnier. It's hot. It's probably hotter than Mercury. It's probably within the orbit of Mercury and just zipping around in there. They also found a sub-Neptune size planet, which is HD 21749b. That one's a little bit further out and is a bit bigger than our planet. They definitely think it's a gas giant, and it has an orbital period of 36 days. I'm wondering at what point we're going to start finding planets. We keep finding these exoplanets that are super close to their stars. We're using this transit method where the planets dip, they transit between the star and our telescopes, and we see the dip in light, and so we're like, something's there, right? The reason we're finding them is because it's this really easy to catch pattern when you're looking at something zipping around a star once a week, right? Or even once a month. You look at the data and you go, it's easy. But when we start thinking about something like Earth going around a star once every 365 days, I mean, it's a much longer period of data collection to actually see the transit and to know that you are getting a replicatable signal, right?
Great.
Yeah.
Just calling you out on your filler word. That's all. No big deal.
Thank you. Right.
All right.
And then finally, last story, there is in the Tatooine system. It's not really the Tatooine system. It's known as Kepler 47, named after the star. It's called Kepler 47. And they have found a number of planets in this binary system. And that's why it's called the Tatooine system, because it has 2 stars, 2 suns that these planets orbit around. If you could imagine being on Tatooine and seeing 2 suns come up for sunrise. They just found a 3rd one. There's another star in there. I mean, not another star. There's another planet in there. So there's more planets in the Tatooine system.
But do they have blue milk is my question.
I don't know if they have milk at all.
Oh, well. If they do, it better be blue.
It should be.
I'm gonna be pissed.
Yeah. Anyway, new planets, exoplanets. Is there life out there? Possibly. That's the final story for that.
Probably.
Yeah. What's up with you, Justin? What you got there?
Let's see. Where am I at in the rundown of my show? Oh, sometime around 13 billion years ago, the universe was a simple soup made up of 3 simple single atom ingredients. That was it. That's all there was. They existed in a rather dim universe, as stars were still not going to be invented for another 100 million years. But within that first 100,000 years after the Big Bang, the very first molecule was born. A soupy love child of helium and hydrogen known as helium hydride ion, HEH+. Quoting voice of David Neufeld, professor at Johns Hopkins University and co-author of the study, which scientists have finally detected this elusive molecule in space. It was the beginning of chemistry. That's how big a deal this is. Theoretical models have convinced chemists and astrophysicists that HEH+ was the first molecule, followed in a pretty precise order after that of a parade of increasingly more complex molecules. We have actually been able to study this in a laboratory. It's over the last 100 years in laboratory settings, but have not yet detected it in its natural habitat. Which then, you have this pretty convincing idea of how everything works and how everything would have been put together, but you can't see it. Why can't we see it? So part of the problem, it turned out, was they knew where to look. They thought, okay, we should see this in glowing gases being ejected from dying sun-like stars. Create some sort of conditions similar to those found in the early universe. The problem, though, was the electromagnetic waves given off by the molecule were in a far infrared range, which became completely canceled out by Earth's atmosphere. So it's undetectable from the ground, which sounds like a good alibi for why your hypothesis or why the theory itself isn't working out. We just can't see it. It must be within this wavelength. So I don't know how they figured they did this. NASA and the German Aerospace Center joined together to create an airborne observatory. They had a 2.7 meter telescope, an infrared spectrometer, and a Boeing 747. They cut a big window in the top of the fuselage, and they flew their observatory to about 4,500 feet.
That is awesome.
Yeah. let's just take the lab, put it on the back of a plane, cut a window in the top, off we go. Yeah, so this is the Stratospheric Observatory for Infrared Astronomy, or SOFIA. Managed to get up at that height. They were actually able to be avoiding 85% of atmospheric noise that ground-based telescopes would have to deal with. And data from a series of just 3 flights back in May of 2016 contained the molecular evidence that they saw. So now that they've got it sorted, yeah, sort of an interesting thing is it's the universe in the early stages around this time, rapidly after the Big Bang, was still about 4,000 degrees Celsius. That's a hot ball of just atoms floating around. So you can kind of see, like, with that heat, you would expect those atoms to be moving quite a bit. But even then, this bond is very fragile. So the fact that this molecule even got started and then progressively was replaced by larger, more complex molecular bonds, maybe this whole universe is just a freak of probability.
It probably is.
Probable.
Probable.
If it's probable, it's
probable. Absolutely definitely probable. Moving on to the next story. What did I have coming up? Oh, AI. This is a quick love story to artificial intelligence and what it is going to do for us in the future. So, there have been a bunch of warning stories about artificial intelligence and, oh my gosh, the world's going to be taken over by artificial intelligence. What are we going to Don't trust Facebook, don't trust this, and Siri, and what are we going to do? The robots are coming. But in some cases, it's very helpful and actually really good.
Some cases.
Justin, you remember years ago when folding at home got started.
Fold it.
Fold it. There's folding at home,
Exclamation
fold it.
point.
Yes, it is a crowdsourced citizen science effort to match up protein folding sequences with reference sequences that have already been identified to determine whether proteins actually fold the way that we predict them to. And for the most part, we still don't do a very good job at predicting how a protein is going to fold, especially when it comes from a sequence of DNA or RNA or originally that is several thousand base pairs long. So shorter sequences, we're getting it down. We're able to understand based on basic physics principles, angles at which chemical bonds take place,
Got your
and at which
hydrophilic, your hydrophobic,
exactly how
hydrogen
things are
bonds,
attracted to
all
each
these
other
things.
or not. And there are many there are very set rules. We just don't necessarily know how all of them play out in this more complex uh, environment, especially when you take a thousand, 1,000 amino acid base pair long uh, strip of DNA. And then it folds to its primary structure. And then it combines with maybe another chunk of protein from somewhere else. And suddenly you've got this massive tertiary structure, and things are bonding to each other and refolding all over the
There's
place and
bonding, and then
adjusting
there's
to
these
each other.
activation sites, these docking
Yeah.
ports. This is how all enzymes work. That's just
When things
totally
interact with
right.
those, then it changes the
If the
structure of
and
the
then that
whole.
suddenly the enzyme doesn't do the thing it was doing before because the docking
Yep.
ports are different. These are just a fascinating universe
It's
of
fascinating.
things we would love to know.
We
Huh,
would love to know how proteins fold, and so this
too
effort
bad
is
we never
now
will. Oh, wait, what?
it's now being undertaken through artificial intelligence, and previous efforts with artificial intelligence have used codes, programs that are thousands and thousands of lines of code, and they're very the code itself to run the program is almost as complex as the proteins that we're trying to unfold and figure out how they work. And so these AI programs historically and the current methods that we use are time intensive, and they don't always give a very accurate representation. Well, a researcher from let's see where is he from Harvard Medical School, Mohammed Al-Qurashi. He has reported in Cell Systems this week about his artificial intelligence system, which is taking deep learning, in which you take known sequences and you train your algorithm based on the known sequences, and you do it over and over and over again and get results. And this deep learning is giving similar results to what is out there currently. The difference is that it takes minutes instead of days to weeks for the processing time.
And this is like one of those things that I'll be disproven on again one day into the future. But the thing with the deep learning is every deep learning thing has come up with available answers. Like the deep learning coming up with an available answer because the filters that were able to be put in were by known answers. And this is like part of the problem I have with the deep learning. It's like, okay, you put in the Encyclopedia Britannica and you asked it for a this an animal with stripes, and it came up with tiger. Like, but you put in the information to begin with. So I'm very skeptical of deep learning still. I don't feel like it can give a new conclusion yet. I haven't seen the deep.
So where it is, and instead of that basic layer of the human processing, like with Folded, what you're doing there is you're using human processing power for recognition, pattern recognition, to be able to make the same kind of matches that this algorithm, this AI program would be able to make. And what the AI program is able to do is increase the complexity of what is observed at any one moment in time. And so the depth of what this deep learning can potentially do is potentially that it can go past the human capability for pattern recognition and these big data sets for these big structures, that it will see things that people can't, that it's going to go beyond us. And that's what's happening.
I think it can find the needle in the haystack quicker, but it's still going to find the same needle that humans would have found. I guess I don't see it yet as a thing that can do new learning. I guess that's what I'm
okay. It's not new learning. It's what people could do, but now where it was slower previously, now the computer programs have gotten faster, and they're faster than us. And that's that's the advancement.
And that's cool.
It's going to make us more efficient. So we will make the discoveries more quickly. And then on the other line of another artificial intelligence deep learning application, not just to protein folding, but to fusion power. We all would love limitless fusion power, wouldn't we? For cheaper electricity, limitless electricity. It would be a wonderful thing to eventually get to that technological stage. And the technology that is on the forefront of that right now are tokamaks and ITER, the French and European Union collaboration, which should be coming online. I don't know what year. It keeps getting pushed off for many reasons. The issues they come up with are disruptions in the plasma field, the plasma generated within the tokamaks. And those disruptions can not just damage the fusion process that's taking place, but also damage the tokamaks themselves. And so people who are running these tokamaks, they're constantly having to monitor what the plasma field is doing to keep these disruptions from taking place. And humans do an okay job of it, but artificial intelligence can do it better. And how much better? Well, they're getting into predicting disruptions, and they're using deep learning to learn from model tokamaks. They're currently using the Joint European Torus and also the DIIID National Fusion Facility at the Department of Energy in California at General Atomics. They're using these to model fusion reactions, take that data, and then predict what will happen in tokamaks that are being constructed and in energy runs moving into the future. So the idea is that we will have better control over these energy fields, know when these disruptions are going to be coming up before they happen, so that they can be taken, the disruptions can be disrupted before they disrupt things.
Very meta.
Yeah, sounds very snake oily too. This is my new other things that I have predicted against in the past. I've failed. Some of them I'm still hanging in there. This deep learning thing. By the time they did the next generation of this is like correlative data to me. It's not new data that they're going to come up with. It's not really going to be that powerful thing. The next thing that comes after that, they're going to name it something else, and that'll be the thing. That might be cool. It might be built off of this. But the deep learning as it is now, I have not been impressed with yet. It's a lot of people being like, look what this might do. Eventually. I'm not seeing, like, give me results. Show me that this thing's like,
That's how technology works.
yes, it is. I do understand this, but
We're-
it's just, I'm not impressed with it yet.
You're
You're
impatient.
not
You
im-
want the deeper learning. That's
want-
what you
No,
want.
no.
You want
You
your
want the
learning to be
results.
super
I want
deep.
now discovery
Mhm.
AI, not help you figure out a large data set and give you the answer you got through a harder way. Before, like, I don't even know if it's doing that yet. Like you said, the code to write it's more complicated than the thing it's trying to look at.
No,
And
but
it's
so
not seeing it yet. Like,
So in-
that's a problem.
No, so in that AI program I just talked about for the protein folding, they've improved the code so it's simpler, it's faster, and it is an improvement. And they are doing this all over the place. The improvements are happening more and more rapidly, and we are going to see the influences of deep learning on the pace of development of the technologies.
Now,
that
by the
affect
time you
us.
see those improvements, I'll be on a different town telling them all about the wonderful benefits of. And
There
we'll
you
see.
go.
We'll see. We'll see.
Okay. All
I don't know
right.
why this one is a
I
bug
don't
in my
know
head.
why. It is a bug in your head, but I see the potential.
Mm-hmm.
I'm optimistic that way. Oh, I'm also optimistic that we are at the quick stories section of the show. I've got some biological machines for you. Researchers from Cornell University are using what they call DASH, DNA-based assembly and synthesis of hierarchical materials. These DASH materials are being used to create lifelike behaviors in materials. Kind of like a slime mold, in a sense. They are using
I'm listening.
yeah, they're using DNA to self-organize and also create locomotion. And there is a system that manages uh, metabolism. So these cells have they're not cells, but this material that they're creating, this dash material that they're creating, has metabolic function in that there is energetic need and waste is swept away and there's form and function. It's not alive. It just acts like it.
This is how we get the brave little toaster in real life.
Maybe.
It wasn't a cartoon. It was a horror story from the future about.
I don't get
toasters
I don't know.
a lot. You don't know? You're not familiar with the brave little toaster? Oh no,
never
that's.
heard of it.
I don't need horror
Yikes.
stories about toasters, but
This was a toaster that was alive, and he had a friend that was an electric blanket, and a friend that was a lamp, and a friend that was a vacuum. It's it's assemblage from my very particular niche of childhood. There's about
I don't think any other parents bought that book. Like, you're sharing
no,
it
it's
like,
a film.
like,
It's
you
a film,
know,
and I
like
guarantee
the
you,
Gingerbread Man or Hansel and Gretel
people
or this
born
living toaster my parents.
people born mid 80s to like 1990 probably are very familiar with the brave little toaster.
we'll
Tweet
have to
at
do a
me,
we'll
people.
have to do a scientific study of that one.
Yeah, because
Anyway,
I've had 3 kids since then. Never came up.
No.
So whatever it was,
it's not
they
good enough
stopped
to reach
printing it.
back to by any means. Sorry
Okay.
about that.
who's up next? Who's got a story
I'll go
for me?
since Justin was talking earlier about the primordial ooze. I have another story about that very same ooze from MIT. This showed that shallow bodies of water were probably more suitable for Earth's first life. Researchers report that shallow bodies of water on the order of 10 centimeters deep could have held high concentrations of what scientists believe to be a key ingredient for jump-starting life on Earth, that is nitrogen. In deeper oceans, nitrogen has a harder time establishing a significant life catalyzing presence. If you think about the origin of life requiring fixed nitrogen, then it's tough to have the origin of life happen in the ocean. It's much easier to have it happen in a pond.
Life came from ponds.
Yes, so there's all sorts of things happening here with RNA and lightning strikes and rust. It's very, very, very interesting. I suggest you check out the show notes. But ultimately, if nitrogen's involved with the origin of life, the water, it appears, would need to be shallow.
All right. Good to know. Justin, tell me about
You
ancient.
say you want to be an archaeologist, but you got no bones, no tools, no artifacts of any kind. No problem. An examination of soil layers in an early Neolithic site in Turkey reveals that they are highly enriched in sodium, chlorine, and nitrate salts that are most commonly found in human, goat, and sheep urine, offering then a kind of a signal for us to start following the management of those animals at this site throughout history. The findings, along with, they also had some enriched nitrogen signal in the soil, as well as higher soluble nitrogen levels in trash heaps, which we actually even see in modern feedlots. They were able to, this is Jordan Abel and colleagues used several techniques to identify the urine salts and to distinguish them from natural geological salt deposits. And they found that 5 to 10 time increase in these salts between about 10,400 and 10,000 years ago, and a 10 to 1,000 time increase in the time between 10,400 and 9,700 years ago. So they could actually track a switch to herding and possibly a domestication event in animals over a 1,000-year period without the typical archaeological tools necessary to do such things, just finding the traces of urine that were left behind by the animals at the time. Very interesting. And they say they were only required a population of somewhere under 2,000 people. And then my last quick story tonight, this is one that seems like has come up before. There was an interesting thing off around Monterey Bay a while ago that was looking at there was an event where there was a orca that killed a shark. And what was interesting is the sharks in the area that weren't attacked disappeared from the area, and they like across this whole western seaboard, they all like took off all at once, went to that deep ravine out there in the middle of the Pacific Ocean somewhere, didn't come back for a year. And the idea was there was this scent of this killed shark that had scared them off because the orca had killed it, which for an apex predator like the great white shark makes an incredible amount of sense. You're an apex predator. Something out there just killed one of you. Let's all just leave. It's a nice defense mechanism that you wouldn't expect from something that we consider to be an aggressive predator. So this is a study that in Nature Scientific Reports that showed that when confronted with orcas, white sharks immediately vacate preferred hunting grounds and do not return for up to 2 years. This isn't even showing a kill. There may have been a kill that took place somewhere that they didn't see because this is all happening under the ocean. But they noticed it's just a passing group of orcas is enough for sharks to leave a feeding ground and to stay away for an entire season. The sort of the interesting thing is the orcas, they might just keep going. They're going to some other hunting ground further away. Monterey Bay Aquarium noticed this, and they found that the beneficiaries of this, the ones who are really, you would think, the sharks, of course, because they're not having to confront orcas, but it's the elephant seal colonies, because the orcas go by, they weren't there to feed on them. The sharks that would have been there all left because they were scared, and now it's just open, safe waters for the elephant seals for an entire season. So orcas helping seals where they can. Sharks afraid of orcas.
Who would think it? That's wonderful.
Yeah. Oh my goodness.
You wouldn't think great whites would be afraid of much.
Oh, it makes sense. If it's going to be
does.
anything, it's going to be an orca for
The
sure.
orca's the only thing out there
The
that
orcas
can.
are going to be way smarter than those sharks anyway.
They are smarter, and they have hunting techniques, because that was the thing where you flip the shark upside down.
Mm-hmm.
That was the attack that was witnessed that led to the sharks all taken off from the whole West Coast when the orca killed the great white off of Monterey Bay. There was footage of it. Somebody had caught it, like people who were out whale watching or whatever, caught this on video, and the orca hit it and flipped it and held it upside down as it pushed it through the water, which incapacitated the great white. That's an intelligent attack maneuver. And that's what's funny is they also do it on the stingrays, which are related to sharks, which also, if you flip upside down, go into that catatonic state. Pods of orcas have been seen utilizing that technique on them too, but they don't utilize it on anything else. They figured out
which
what
ones
it
they
works
need.
on.
Yep.
They don't go flipping everything upside. Everything I get into, I'm going to flip it up. No, it's just the things that go into the catatonic side. They've learned this, and it's a passed on knowledge. It's a really fascinating element of orcas.
And before we put everybody into a catatonic state, no, that's not what happened while you're here. It is the end of our show, though.
Well, maybe not. Okay.
It is time for us to give those shout outs and thanks to everyone. Thank you to Fada for helping with show notes, YouTube descriptions, and social media. Thank you to Identity 4 for recording the program. Thank you to Gord McLeod, Ben Rothig for moderating in our chat room. And thank you to our Patreon sponsors. Thank you to Paul Disney, Richard Onimus, Ed Dyer, Andy Groh, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K. Bob Calder, Time Jumper 319, Eric Knapp, Richard Brian Condren, Dave Neighbor, Aiden Jeff, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Corinne Benton, Sue Doster, Dave Wilkinson, Ben Bignell, Richard Porter, Noodles, Kevin Reardon, Christoph Zucknerek, Ashish Pants, Ulysses Adkins, Sarah Chavis, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slazewski, Jim DePoe, Greg Riley, Sean Lamb, Ben Rothig, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for your support on Patreon. And if you are interested in supporting us, you can find information at TWIS.org. And remember that you can always help us out by telling your friends about TWIS. On next week's episode, Jonathan Foley from Project Drawdown will be with us post Earth Day to talk about Project Drawdown, what it is, what are the solutions that are out there for us when it comes to climate change. And we will be broadcasting once again live online at 8 p.m. Pacific time on twis.org/live, where you can watch and join our chat room. But don't worry, if you can't make it, you can find our past episodes at our YouTube channel. Look for This Week in Science on YouTube or twis.org.
Yes, thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have one of the mobile type devices, you can look for TWIS, the number 4 droid. And is that one still there? We should look that up. Somebody told me the TWIS 4 droid app isn't in the Android marketplace anymore. Or This Week in Science in anything Apple marketplacey. That you should still be able to find.
For more information on anything you've heard here today, show notes will be available on our website. That is definitely still working. That is www.twis.org. That's TWIS.org. Where you can also make comments and start conversations with the hosts or other listeners while you're there.
Yeah, you could. Or if you don't feel like being quite that social, you can just contact us directly. Email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in your subject line. Otherwise, your email will be spam. You can also hit us up on the Twitter, where we are at TWIS Science. That's with one S. At Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there is a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, right here. And we hope you'll join us again for more great science news.
And Andy Groh, if you've learned anything from the show, remember,
it's all in your head.
in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This week in science. Science, science. This week in science. This week in science. Science, science. I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This week in science. Science, science. This week in science. This week in science. Science, science. I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
All the buttons to push and all of the settings to set, and sometimes I miss things, but I got it. Thank you for watching the show, everyone. Thank you for joining us for another episode of This Week in Science. Anyone out there live near Santa Fe, New Mexico? Anyone live near Santa Fe, New Mexico? I cannot stay much longer tonight. I have a child who did not go to sleep when I asked him to. And he's still awake and he's clearing his throat. He tells me he loves me.
You're never gonna love me.
Oh, I love you so much. I just think it's the wonderful strategy of the child. You're mad at me. I love you. I love you. It's a wonderful strategy. It's great.
Sorry, I just started a Twitter poll about the brave little toaster. So
Oh, you did. Oh, good. You must share the results when they are in.
I've been shouting on the internet today. I hope everyone appreciates it. At
So
Shouty
much-
Blair.
So much shouty Blair today, huh?
I did some shouting.
Oh my goodness.
Here, I should retweet to my other Twitter so that people can see it.
Oh, Identity 4 has framed the kiwi bird. Let's take a look at that. Gotta open that up. Hold on. Oh, looking good there.
Oh, hey, Identity 4,
Let's
please
share
social
this.
media that so that I can share it. Please, please.
Look at that. The kiwi bird. Over to mixers. What's that? I got destinations. You got UFOs? No, LFOs. LFO1, LFO2. Destinations. Look at that. Little framed kiwi. That's cute. You can have a kiwi tote bag. They're on Zazzle. Mhm. People out there. Wow, awesome. I'm glad. That was hand delivered for the first time by Blair, who brought the kiwi to Portland, and Identity came down to our live show. He's up in Seattle. That's so great. And it's sitting above his synthesizer in his music.
How fun.
studio. Very nice. Very nice. Yes. Where'd Justin go? He's gone for a long time again.
Instead of a single cigarette, he's smoking an entire cigar.
I know, something like that.
Swirling
my
some
goodness.
whiskey.
Oh, good. Ellsworth Danby in the YouTube chat room says that our podcast is on the Android Player FM app. Great. So glad to know that. We're out there. We are on all the apps. I know we're there. We're out there. We're kind of like, the X-Files. Well, what the X-Files were looking for. It's out there. We are out there. I'm tired. My child is awake.
I'm tired too. I hit the snooze button 6 times this morning.
Yeah, I had a hard time waking. I did not go to bed late last night, and I don't know what happened. I woke up so poorly this morning.
I've been waking up bad all week.
I think the spring allergies are starting to hit my sign. I woke up and I was like, my head, my sinuses. Oh my goodness. Oh my goodness.
Yeah, I did not think that I had allergies. I still am like, I've never had allergies. But I'm starting to have to give in, I think. One of my coworkers today said, I didn't have allergies a day in my life until my 30s, and it hit my mid-30s.
Yep.
Oh.
Ed, an alarm clock to wake me up and a Kai alarm to put me to bed.
That's like a car alarm.
It is a Kai alarm. Oh, Hot Rod, you have itchy eyes. That's the worst. I don't like itchy eyes.
Is that the Kai alarm is sounding over there?
Yeah, did you hear the Kai alarm sounding? Yeah. So knowing that you can hear that,
or a
he's ringing. I don't know. He's just ringing. I
ringy
wonder.
dingy.
Well, he was loud tonight, wasn't he? Crinkle, crinkle, crinkle.
I heard I thought
Crinkle,
it sounded like somebody typing, but neither of you had your hands moving. So I was like, ooh, a ghost is typing.
Yeah. I don't like cleaning audio.
It wasn't bad. It was just a couple times.
All right. Eric had a friend who developed an allergy to walnuts in his 60s. No, you're not supposed to get allergies that late. That is
especially
frustrating.
food allergies. That seems weird.
Right? I know. Yeah, and Identity 4, all them grasses and trees getting busy all over my face. That's right. Yeah. All the pollen. Oh, the love of plants. I have allergies.
Yeah.
Yeah, thanks, Chris Harden. Chris Harden is sleepless in Lansing, Michigan. Yes, you are. You're up late in Lansing. My goodness. Kai, are you going to get a PhD?
I'll try.
He'll try. He says he'll try.
I'll try as hard as I can.
We'll see. What's your career you want to do right now? What? Oh, his new career, he's going to be a global warming protester.
It's a career, eh?
I didn't know that was a career.
You start saving now.
A scientist, astronomer.
I mean, there's a way to be a global climate change
Wanna be an-
protester that makes
be an-
money, and that is
Wanna
to be an educator.
He's going to be a marine biologist. That's what he says. He says he'll be a marine biologist.
Sea spiders. Study some sea spiders.
Oh my goodness. Yeah, he could study sea spiders. He could be the intern that pushes the spiders over.
Yeah, that would be so fun.
Good night, Adam Weaver.
I'm not a jerk. I just think it'd be very fun.
It came off as very jerky.
Oh
It did.
boy.
You were encouraging spider tipping.
No, it's just funny that you're like, this is how I'm gonna test exhaustion.
Hey, did
It's
we?
like, I mean, you can't put them on a treadmill. I get it.
Did we
think
ever
it's funny.
explain the Andy Grow thing to the
No,
minions?
no, but
I
that's
kind of
cool.
like that it's left unsaid.
Yeah,
too.
because
leave it unsaid.
Okay.
Yeah.
Okay.
maybe
Leave it
that's a secret patron level that people don't know about.
Secret. It's a secret. Charles Abroad. Yes. Did we grow up in the same town outside? I did not grow up in Lodi. I grew up near Linden, and I went to Linden High School.
Same
So close. It's not the same thing. It's close,
rural
though.
Central California. You had this same experience.
Yeah, we were. Yeah, it was like 20 minutes apart,
Yeah.
30 minutes apart. Yeah.
Hot, dry, and well,
boring.
Boring is an understatement.
Oh, no.
They're trying to
How
get boring to
sad.
this day.
What do you do with yourself? Oh, I don't know.
Oh, how sad.
I grew up
That's
out in the
before
country.
there was even a Walmart to hang out in.
There wasn't.
Wasn't even that.
Yeah, we grew up. Charles Abroad. Maybe you remember cruising on Pacific. Was it Pacific? Was it Pacific in
So far from
Stockton?
the ocean, by the way.
Yeah, no, not
No,
the Pacific.
No ocean breeze makes it to
No.
Linden.
No, I remember doing that a couple times. Oh my goodness. Oh, high school.
Oh, town with 4 stoplights.
Right.
Yarks.
Was it
That
4?
just
Was it 4 at that point? Or did that come later
sounds
in Linden?
I think the stop signs came
upsetting.
later.
The
Yeah.
stop signs even came later. Like it was
Yeah.
not even, they didn't even have
No.
markers.
I don't even think they still have stop lights. It's just they have a
Oh.
few stop signs.
I'm not
Yeah.
making fun of your town. I actually would love that. Maybe not the hot, dry, arid desert of the Central California Valley, but
No.
yeah.
Come on. You grew up near there.
Me? No, not even close.
Oh, no, you didn't, did you?
No, not even close.
No, it was just later that you ended up there
What do you mean?
in Davis.
Oh, and David. Yeah. Well, I was here. Yeah, David, but that's not the. You cannot compare the arid desert to the Delta.
That's true.
The
You're close to the
Delta's-
Delta.
Yeah.
But no, I was kind of close to the Delta. I mean, outside Stockton has the Delta go right through it.
Well, yeah. But you were outside of Stockton.
I was outside of Stockton, where the walnut trees and cherry trees grew in groves, offering plenty of shade.
And that's what we did for fun. We cooled off in the shade.
Growing up, I mean, honestly, I swam in ditches. I didn't have a pool. I swam in a ditch.
Oh my god.
That's awesome.
And my dad had at one point somehow he ended up with my cousin's surfboard or something. And so we'd go up and down the ditch on the surfboard. That was like summer entertainment.
How much tetanus did you have?
That's very weird.
Oh, plenty.
That's very weird.
That's very weird. Pipes and Kai. Yes, Kai.
Kai
you
thinks
have
it's
a
very
charming-
weird. Exactly. Yes, it was Pacific Avenue. Thank you, Charles.
I'm halfway living in a ditch.
I didn't live in the ditch. I didn't build a house on a bridge over a ditch.
Oh my gosh.
Your imagination is something else. Okay.
So I'm gonna. Can I segue into a little childhood story? I don't know if I
Segue.
told this one before.
Segue.
So my summers were spent up in the high foothills, low Sierras, and there's one summer, I was 7. I was there with my 4 cousins, the oldest of which was 11. And me and my cousin were talking about this one summer that was just amazing. It was totally like the Tom Sawyer freedom. We were out on this 800. We were kind of home caretaking an 800-acre ranch. And I remember we went out and we hunted bullfrogs, and
Mhm.
and I remember we even like had to kill a rooster. We were milking goats. We were doing all this country bumpkin just fantasy life. It was just fantastic. We had horses.
Fantasy life.
We had
Yeah.
horses from the Bureau of Land Management because they were giving away Mustangs because they're relocating them from Nevada.
Oh, that's
And
cool.
these things, my uncle
Someone
would
had
get
to break them.
my uncle would, and by the point they would take a saddle, that was the okay for us kids to ride them. And these were not trained horses. These were like half wild horses still that would sometimes take off on us. But I'm recounting like all the fun and the adventures we had, and we made our own raft to go out to catch the bullfrogs, and we built our own thing to get them with, and had this whole technique where 2 of my cousins would rattle the cattails and the bullfrogs would come out across the top of the water, and me and my other cousin were out there on this raft and we'd catch them. And he's like, "You know why we were doing that, don't you?" "What do you mean?" "Yeah, it was fun. It was awesome. We were Huck Finn, Tom Sawyer, and it." Like, no. Our uncle, who was a miner, he would put the explosives in the holes of the mines.
Yeah, MINER.
Yeah, took
Not OR.
like a 3-day job that got the road got washed out. Maybe I'm trying to be kind. For some reason, he was there 10 days. The pantry that he thought was sufficient, plus the chickens laying the eggs and the goats with the milk and everything.
They ran out of food.
Kids will be fine. We ran out of food. But what I saw is an idyllic childhood romping around. My older cousin, who's the oldest amongst us, who at 11 was like, "No, we were surviving." That was subsistence survival, Lord of the Flies style, that we had to do for like 4 days of that. And then as we're talking about it, I was like, too bad we can't send our kids off to Uncle. like that. There's some lessons learned there that you don't normally get.
You can send your child off to that kind of idyllic experience without the potential starvation.
That's the thing, though. That's the part of it that that's the part that really forged us cousins early on. Is that we really figured out what we were made of early, thanks to Uncle Phil going, "Yeah, they'll figure it out.
They'll figure it.
They got the eggs and the goats. Yeah, they'll figure out the rest of it."
That's funny. Wow, that's a good story. You haven't told that story before. That was good.
Oh my goodness.
Oh, childhood.
Oh, childhood memories.
Childhood memories. All right. Business.
Ooh.
Blair and Justin, I need your traveler information. Send it to me so I can send it to people.
I need the days I have to take off of work.
I will tell you. Everyone out there, the TWIS team is going to here. Let me put it up. I'm going to put it up on the I'm going to put it up on the screen. Hold on. There's a new thing that just confirmed today.
Little side thing that we're also doing when we're there. Meow Wolf.
What's that?
What's that?
Meow Wolf is the ginormous art installation that you wander through, and it tells a story of sorts, but you're literally interacting with it's like part puzzle rooms, part art installation. That's what we're also going to do. But this is
How about a hot air balloon also, though?
We can do that here. Why would we go to that?
Santa Fe's where that's a thing.
It's a thing here too.
No. It's much more of a thing there.
Vacaville.
It's a thing.
All right.
Okay, but there's this thing that the
Hotter
TWIS team
balloons.
is now
Hotter
confirmed to
Oh,
do.
and
We are going to be the podcast and the podcast stage
what?
at the Interplanetary Festival in Santa Fe, New Mexico.
Is this gonna be 3 days of podcasting? Because I love it when we
No, we're gonna be just
Oh.
like one day. We can do more if we want, but as far as I know, it's just
One
gonna
is
be
good.
like one
Well,
show.
one for
We'll
the
just
show,
do one
but
show.
okay, but one for the show. But the other thing is, this is one of those opportunities to grab a ton of really awesome guests to
Yep.
bring on for 15, 20-minute interviews.
Yep.
So aside from the regular podcast, I think we should have our ears up for grabbing lots of people unsuspectingly before or after they've done their presentations and
Mhm.
getting them to talk to us about how awesome their research is.
Yeah, absolutely. We can run around and do that.
Friend of the show, frequent guest, friend of the show, Sean Carroll will be there.
That's right. Yes.
Question. Is this gonna be we're gonna do like a normal show there, right? Like it's
Yeah.
gonna be a normal thing. So
Yeah.
is it gonna be a bonus show, or are we gonna do it in place of a show on one side of it?
Well, we can figure this out. I like the idea of doing because it's not going to be covering our regular time slot, right? This is gonna be
No, it's gonna be probably we can choose our time slot, and they've given us the opportunity to pick when we wanna do our show, on Saturday, sometime between, I think what did they say? On Saturday, and it's sometime between, like, noon and 7 or noon and 6 or something like that,
So-
and
Cool.
on June 15th. So we'll figure between noon and 6, we're gonna be in this Innovation and Ideas Expo, and so we can
what
kind of
I
pick where we wanna be.
what I think might be nice, and maybe this isn't the best idea, so I'm gonna say it like it's a question.
Mhm.
Is if we did a show where we're bringing stories that are maybe beyond this week, but are themed to
Mhm.
the event.
I think
Like
that would be fun. Yeah.
kind of like a
That's what I
rundown
was
of
kind of
all the
asking,
amazing
yeah.
things, maybe just the last 6 months or year, in space exploration.
Yeah, I mean, seriously,
Really
we are gonna get an interview with somebody from the Santa Fe Institute, so I'm talking with them about there would either be, what's her name? Gosh, Jennifer Dunn, who she researches, complexity of ecosystems, and so it would be the idea of ecosystems for space travel and how could we put people into outer space and what do the ecosystems need to be like, or the head of the institute, David Krakauer, who does other spacey complexity kind of stuff, and so we are going to have at least one interview, which will be great.
yes, that would be awesome.
Yeah, but I like the idea of kind of theming it up along with this interplanetary,
Mhm.
what's yeah.
How long are we doing? Are we doing a 90-minute thing? We haven't decided yet.
We haven't decided.
Do we have a time limit?
If
I
there are
feel like
if there are other people in the schedule, yes. Yeah, we have to, we have
because
to
we said we could
it
pick
will
our
be
time
a set
So
thing.
it sounds like it's not the dance card isn't quite filled out yet. My
It's not,
point though
no.
is that we could do a 60-minute show, a 30-minute show where we're talking stories of recounting a lot of the awesome stuff that's been going on in the last maybe 6 months. Even doesn't even have to be a year, but also we could fill another hour with 15-minute interviews. I like
Mhm.
the idea that whenever we're at one of these events, we have a sort of shooting fish in the barrel opportunity to talk to a lot of these people, and
Yeah.
I would not want to waste that opportunity.
Yeah. Yeah, exactly. I think it'd be great, or we could, run around with a camera and do interviews or something. Yeah.
If we have a stage and mic set up and sound check already done, why not just
Yeah, so anyway, the whole thing looks really fun, and it's Santa Fe, and I've never been to Santa Fe, and I'm very excited, and it looks like it's just gonna be a blast, and
Mhm.
yeah, so, we'll be going there on the 14th. They would like us there by the early evening on the 14th.
Mhm.
And then we can go home Sunday, Monday, whatever works for us. Yeah, but that's where we're gonna be in June. June festivaling in Santa Fe. Extra show. And if it seems too crazy, we can be like, maybe that'll be a show.
Oh, Planet of the Apes is a keynote. Oh my gosh, this is going to be very interesting.
Oh yeah, no, so the keynote they're doing what did they tell me? Yeah, it's going to be this guy J.W. Rinsler is going to introduce the screening. So I guess he's a writer. Oh, he wrote a book, The Making of the Planet of the Apes. And so he's going to give the keynote about the movie.
Interesting.
Yeah, so this festival brings together science and space and art and music. And it looks like it's going to be a lot of fun if anybody wants to go to Santa Fe. Yeah, and Sean Carroll's going to be there. Oh, and also the authors Daniel Abraham and Ty Frank, who've written the Expanse series, they will be there also. I'm excited to meet some of these people.
Yeah, this is
I'm gonna bring tardigrade stories. So there.
You're like, I already figured it out. That's it. Wait, what did you say? Direct flight? So what they suggested we do is here, I'm gonna stop presenting. Stop presenting. They recommended we fly into Albuquerque. And then they said there are shuttles that will take us to Santa Fe. And they said, don't try to go to the little Santa Fe airport.
Okay, yeah.
I just got
They're
that
like, that's not a good idea.
I got that exact same advice when talking about this show from my sensei at work, Crystal. She was like, whatever you do, the way she described it, they're like, the entire airport has one vending machine. That's that's
Lord.
the food.
Okay,
That's the
so
food court
Albuquerque.
is a vending machine.
All right.
Yeah.
I wonder if it would be cheaper to rent a car than grab a shuttle.
No, I think there might be free shuttles.
Oh, you think there's free ones?
Yeah, I think they said something about there being shuttles and that she was like, yeah, just get a shuttle. It'll take you right to your hotel. And
I would rather.
But if
I
we
would rather
land at
take a hot air balloon. Can we do
Yeah.
that?
Of course you
Yeah,
would.
Take a hot air balloon to
You
Albuquerque.
just gotta test the wind first.
That's right. Yeah, so I don't know if we can all try to get to Albuquerque at the same time. We could maybe rent a big vehicle or something, or
Yeah,
get in a shuttle
minivan.
together, depending. Yeah, minivan or something like that.
Wait,
Yeah,
why do we need a
and then
minivan?
we'd be
Oh wait, no, that's right.
mobile.
There's going to be more than just the three of us.
Yeah,
Maybe.
and then we would be mobile around Santa Fe.
Yeah, that'd be pretty
Getting
cool.
places, which would be kind of cool.
And
yeah.
Meow Wolf. I think that's what it is. Meowwolf.com.
And the balloons.
You have to check out this Meow Wolf thing. If you have,
also,
I saw
all of the turquoise you could ever want. That is the New Mexico stereotypes I've heard: hot air balloons and turquoise jewelry.
I heard spicy food and I went, I'm sold.
Nice.
So I was talking with Jenna and her coworker today, and who were the women at the Santa Fe Institute who are running this festival, and they're like, do you like spicy food? And I was like, yeah. They're like, you should live here. Like, okay.
Move here immediately.
That's not Portland. Hold on. That's not what. Hold on. That's good. I want to eat all the food. It'll be great. They told me I'm not allowed to bring rain. Okay.
Huh?
All right.
Does it ever rain in Santa Fe?
So here's the thing. I don't know. I don't quite know how far away it is. Albuquerque, I've visited in February before. There was snow. It's a high plateau. I mean, it's desert, but it's a high plateau. So yeah, they get like roofs covered in snow.
Huh.
I can't wait. I know. I'm looking at touristy things. I'm gonna touristy. Yeah, noodles. I gotta put my head and my child's head to bed also. Tomorrow's gonna be hard to get this child up for school. But yes. We will
Yes.
make it so. We're going to
Yes.
this fun festival and it's gonna be awesome.
Let's all go. Yes.
Yay, let's all go.
Yes.
Yeah. So if anyone wants to meet us in Santa Fe, we'll see you there.
Whoever that might be.
Yeah. Blue crystals. Ooh. Oh, I won't break any blue crystals, Kygo.
Can we pull up the Meow Wolf thing?
I don't know. Meow.
Just Google it and look it up. This is a
Oh,
thing.
there it is. Meow Wolf Santa Fe. There it is. Okay. Immersive experiences. Immersive art experiences. Wait, what? Is there one
It's
of
a
these?
giant installation. I watched a documentary about this thing, and was
Can you
blown
say
away
that
by
word
it.
again?
Huh?
You said documentary.
I was like, it's like a mini documentary. It was like this thing about the making of.
I just really liked how excited you were.
And I was so impressed by this. I was just like in awe of what they had built. And
Santa Fe.
And like you have to like walk through a dishwasher, and it's all Alice in Wonderland. Well, it's not quite, but it's just, and it's interactive and it's insane. It's off the hook, and it's just an immersive museum storytelling experience.
Yeah, you should check out One Dome in San Francisco.
One Dome?
Too far.
I think no.
What? Rude.
Yeah, no, it's called One Dome in San Francisco, and it's this kind of immersive art experience, and a friend of mine runs it, and they've got this thing. It's called the Unreal Garden. So they used augmented reality and virtual reality and some of their stuff.
I will have to check that out. That sounds awesome.
Yeah, it is supposed to be very awesome. But so Meow Wolf sounds cool to you. I'll have to look into it. I don't know what's going on there. That doesn't make any sense. It's immersive. I don't know what that means. Isn't that life? I am immersed in life anyway. I'm immersed.
Yeah, sure.
Take the left turn at Albuquerque. All right, Eric. Thank you. Lots of art galleries. That'll be fun. It'll be such a fun trip. 7,000 feet above sea level. Thank you, Ed. You drove there and it was a semi-terrifying mountain drive. Oh, great. That's funny.
7,000.
It's not that high. It's high enough. That's pretty
It
high.
is. I was in Denver. It wasn't that high.
Yeah. That's higher than I thought. Yeah.
What?
Darn near mountainous.
It's still flat from what I remember. It's just because
high
it's
plateau.
high.
Yeah.
But if you go, but to get up down the plateau, you might that's how a plateau works, I suppose. But yeah, and I have actually have family in Albuquerque, so I might have to figure that out. Maybe I need to get there a little sooner.
Yeah.
A day sooner. Yeah.
We can work that out. If they said just let us know dates and I'll buy tickets.
I'll make some phone calls.
You figure it out. Make some phone calls. Let me know. Awesome. I'm excited.
Yeah.
Also Earth Day this weekend.
It's Monday coming up.
I thought it was Saturday.
Saturday's when everyone's celebrating, but the actual Earth Day is
like
on
a birthday. You don't do it
Monday.
on a work day. You move
Saturday is 4/20.
Yes.
Earth Day is April 22nd.
Oh, I've always thought that it was 4/20.
Oh no, that's a different day.
It is, but I've just decided that is Earth Day, and that's always been Earth Day in my head. Doesn't matter what the calendar says.
That's Herb Day. That's a different.
It's Herb Day. Yeah.
I could care less about that. Let's see. The sea level folks are gonna love it. Yeah, it's gonna be great. I love it.
Sea level.
I know, high altitude. If you're like walking around, hiking stairs,
Oh,
Drinking
yeah.
beer, getting really
drinking
drunk.
beer. That's right.
Yeah, that's gonna be
Don't
awesome.
drink too much. Yeah, but it's not too high. It'll be
It's
fun.
pretty. That's a pretty decent elevation, even for the Sierras.
Yeah.
That means you're up there a ways.
Yeah.
It's over a mile.
It is over a mile.
It's over a mile high.
Ooh, we're almost in space.
That's another 49 miles.
I know. I know.
That's why that conference is there, because you're basically in
It's
space.
because you're closer to
Basically
space.
closer to space. That's
You're
right.
like, oh look, there's Mars.
Duck! Everybody duck! The moon's coming by.
Yeah.
Oh.
Oh no, I ended up on the moon. How did that happen?
How did that happen?
I didn't duck in time.
Hot in the day, cold at
Oh,
night. I
Blair's
can handle
yawning.
it.
You know what that means, everybody. Say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin. Good
night,
night, Kiki.
Good night, everyone. Thank you for sticking with us for another episode, another fun after show. Thank you, Blair and Justin, for another great show. And
Thank you,
let's
Andy
see.
Grow.
Thank you, Andy Grow. And on Friday, I will do my usual Twitch stream around 1 p.m. Twitch.tv/Dr. Kiki if you want to hang out and talk about science with me for a little while. And then we'll have our episode next week. It'll be awesome. Happy Easter, everyone. I hope all is well. Merry science to you
Merry
all.
science.
We will see you again soon.
Happy
Thank
Easter
you.
Island.